Hey everybody! I just want to do a quick little video over how we actually label our materials in our microbiology class. So you may wonder again why we have to label stuff, and the big thing here is just whenever you do something you have to document what you've done, who's done it, and when you've done it. Those are kind of the three big things that we try to make sure that we get on all of the items that we complete in the lab. So when it comes to something like our plates that we're going to be inoculating, we can generally label these directly on the plate. So just to kind of go through an example of that again. So again, my name is Casper Fredsgard. I'm going to go ahead and put down CF for my initials on there. Let's say that I'm going to be putting some E. coli on it. I'm just going to go ahead and write E. coli on here. So now I have who's done it, what is being done. Got to get the date on there too. Today is the 8th, so May 8th, 2023. And those are the three basic tags again that I should have on everything that I do inside of the lab. Now, generally when it comes to plates, there are two more things we like to put on there as well. First of all, what temperature are we going to inoculate this or incubate this at? So I'm going to go ahead and put down 37 degrees Celsius, which is body temperature. That's a pretty standard one for growing stuff. And then the last thing is going to be what type of plate is it that I'm using. So on some plates, kind of like in this image right here, it will actually say what the plate is on there. It has that little written part to it. But if it's the plates that I make myself for our class, it won't. So because of that, we have to write it down on there. And just because this happens to be a nutrient agar plate, I'm just going to put down an NA plate. And at this point, I have something kind of like this. And I am now ready to get started inoculating my plate. Now the other big thing again that we use in our lab is going to be tubes. And really when it comes to our tubes, we just go for the three basic items. What is it? Who did it? What day did we do it? So normally for that one, we tend to use our tape. And again, I got E. coli on there. It is May 8th. I'm the one that did it, so I put my initials on it. And now I'm ready to basically inoculate my little tube. And again, we tend to use our tape for that one. Just a little note, if we are doing slants, again, the ones that have kind of the little bit of slanted auger inside of there, try to put the tape at the top, not where the slant is. Because when you go to score it after it's grown, it might be kind of hard to see if you got a huge piece of tape, especially if you wrap it all the way around where the slant is, and you won't be able to see anything. But really, kind of with that in mind again, that kind of gets us through how we're supposed to be labeling everything. So I'll see everybody in the next video where we talk about how to inoculate stuff.